Hello everyone, I am Karan Kumar, a research scholar at IIT Guwahati in India. Today I am here to present our research work on computational investigations in inhibition of alcohol aldehyde dehydrogenase, in short AAD, in lignocellulosic hydrolysates. In this work, we have reported a theoretical analysis of inhibition of AAD enzymes by compounds present in lignocellulosic hydrolysates, in short LH. As we all know, lignocellulosic biomass is feasible feedstock for biofuel productions. However, in the pre-treatment step of lignocellulosic biomass, for producing biofuel, many inhibitors are being produced as a result of oxidation and reduction of components present in lignocellulosic biomasses. These compounds strongly inhibit the growth of microbes and interfere with the metabolic activity of fermentative hosts. Therefore, this study aims to identify and homology model the crucial AED enzymes that are present in solventogenic Clostridia. Then, to identify the potent AED inhibitors in lignocellulosic hydrolysates and is- optimize their structure using density functional theory. Then, we will identify the most potent inhibitor among all of them for the fermentative host and their inhibition mechanism. In last, we aspire to propose the metabolic engineering targets for enhanced biofuel yield. The software informations that are used in this study are given in the section 4. Coming to the methodology of this work, first we did the literature curation from which we identified a total of 8 clostridial enzymes that play a crucial role in solvent production and also identified 10 inhibitors from lignocellulosic hydrolysates. So in parallel study, we also selected 4 representative substrates to compare the comparative inhibition profile. So there were total 14 ligands and 8 enzymes. The sequence of these 8 enzymes were retrieved from the NCBI database. The analysis was performed and template selection was made based on the blast analysis. Then, as the crystal structure of these selected 8 enzymes were not present in protein data bank, so we went for the homology modeling. The homology modeling in this study were performed using Modeler and iTager web server. Further, the structural refinement of the models produced from Modeler and iTager were performed using Refine Galaxy. And then they were further accessed and validated using different web servers such as ERAT and ProCheck. We further analyzed the Ramachandran plot of these enzymes also. ProTparam web server was used for analyzing the physicochemical properties of these enzymes. The active site determination were done using Prankwave web server. In parallel manner, we created and optimized the structure of these inhibitors and representative substrates using density functional theory in Gaussian 16 software. This produced high quality chemical structures of ligands. These chemical structures of ligands and protein structures were further subjected for the molecular docking analysis in Autodoc software. The docked structure were further taken for the molecular dynamic simulations using GROMAC software, and then results were visualized and analyzed using VMD PyMol, Chimera, and other visualization softwares. Coming to the results, Figure 2 represents the homology model structures of 8 Clostridial AAD enzymes that were selected for this study. These enzymes can be classified into three categories. First one is bifunctional enzymes, which are both alcohol and aldehyde specific. Second category includes alcohol specific AD enzymes, which are BDH, BDHA, and BDHB, while rest all three are aldehyde specific enzymes. The inhibitors that were selected and representative substrate are denoted in figure 3. These structures are DFT optimized chemical structures. Table 1 enlists the binding energy analysis of all 14 ligands with the 8 enzymes. In the table, we can see that the rocomeric acid, cinnamaldehyde, and vanillic acid are the most potent inhibitor that inhibits almost all AAD enzymes. And using the comparative study of the binding energy and the structural bonding information with respect to the representative substrates, we could able to identify the mode of interaction and mode of binding of the substrates which are listed in table 2. The strongest inhibitor for respective enzymes are also given in table 2. The crucial amino acid information that are very much important for binding of the inhibitor to the enzyme, and these crucial amino acids are also play a crucial role in the binding of substrate to enzymes are also given in table 2. The mode of binding can be seen that most of the enzyme follow the competitive mode of inhibition, and type of interaction that were prevailing in all type of enzyme was hydrogen bond. However, the hydrophobic interaction, salt bridges, and formation of pi cation interaction were also seen in some of the enzymes with their strongest inhibitor. In summary, our analysis shows that the minimizing the presence of rocomeric acid, vanillic acid, and cinnamaldehyde in lignocellulosic hydrolysates could result in higher biofuel yield, thus making the AV fermentation economically feasible. The major mode of binding in all interaction was competitive inhibition. This study also suggests a crucial amino acid targets which can be useful for the metabolic engineering to design the robust strain in the future for efficient biofuel production from lignocellulosic biomass. Thus, this study will be helpful in achieving sustainable development goal. I acknowledge Prime Minister Research Fellowship, Ministry of Education, Government of India, School of Energy Science and Engineering, IIT Guwahati, my supervisors, Professor V.S. Molkar and Dr. L. Barbura, Permission Supercomputing Facility of IIT Guwahati. Thank you everyone for your patience listening. If you have any query, you can direct me an email to karan18@iit.ac.in, or you can also scan this QR code for getting the recent update on my research work.